Hello everyone, it's fantastic to be here with you today at API Day Australia. My name is Dr. Dennis Bauer. I'm a government research scientist, professor at an Australian university and an AWS hero. And today I want to talk to you about cloud-based bioinformatics, or how APIs enable global collaboration and really accelerate health and medical research. By the end of this talk, I hope that I've convinced you that digital health really needs strong APIs. We need to build solutions that are larger than the sum of their parts, and for that we need good collaborations. But before we go there, allow me to introduce the research organization that I work for, CSIRO. In particular, the eHealth Research Centre, which is the largest digital health initiative in Australia and worldwide quite unique in covering this full value chain from basic science all the way up to services and technology that is going directly into the healthcare system. The most famous product that we developed is an Onto server, which underpins the National Terminology Server in Australia and England. We also developed the first mobile health app to be used in a clinical trial, CardiHub. And together with Samri, we invented the Total Wellbeing Diet, and its recipes are now on the book bestseller list alongside Harry Potter and the Da Vinci Code. From that perspective, we do have a nice balance between products that people enjoy and products that people need. Today, I want to tell you 3 stories, and each one of those have their own API element to it. The first one is around understanding the genome and how really bringing genomic information into advanced clinical use. The second one is around manipulating the genome and ultimately coming up with more advanced therapeutics for the future. And the last one is around how we do collaborations, how we can empower innovation from the academic system to flow into healthcare. Let me start by saying that everyone has mutations in their genome that should inform clinical care, be that adverse drug reactions and what kind of drugs should be avoided, to targeted cancer treatments of looking at a patient's genomic makeup of their tumor and then deciding what kind of drugs that tumor is susceptible to. With this wealth of information encoded in the genome, it's not surprising that it's used more and more in the clinical practice. But the genome is very large, and in order to really identify the secrets in the genome, we need to have multiple of them together in order to analyze over those, creating so-called mega-biobanks. So in this era, where we're treating mega-biobanks and huge volumes of data, it's really complicated to analyze that data or even move it around. Therefore, the analysis needs to go to the data, and APIs are perfect to broker that connection. APIs can be the interface between the data and the analytics. They can provide value-add tasks, for example, checking or modulating access privileges. And they can make large datasets manageable by compute-intensive analytics by handing out the data in smaller chunks. Applying this principle, we developed VariantSpark, which is a framework or a platform for analyzing genomic data. On the VariantSpark platform, the data is located on a cloud bucket, an S3 bucket, and is accessed from an elastic MapReduce cluster, a Spark cluster, from within the cloud platform. The reason we are using a Spark cluster or distributed compute is because the data is so large. When you think traditionally about high-performance compute, the tasks were compute intensive. Therefore, all the information would be fitting into one compute node. But now the tasks are data intensive, meaning that the volume of information does not fit neatly into one single node. It spans the boundary across nodes on a regular basis. Therefore, the boundaries between nodes need to dissolve in order to have the data and the information free-flowing. And for that, distributed computing is perfect. So with VariantSpark, we demonstrated that we can process 1 trillion genomic data points, which makes it 3.6 times faster than current application in the space, and it can scale to tomorrow's data, where it can process the data in 15 hours rather than 100,000 years what other technologies would use. We applied this awesome compute power to the biological space to identify disease genes for cardiovascular disease. Here we accumulate a 50,000 case control cohort with people with cardiac disease and without to identify the genes that are driving that disease. With this information, treatments can be personalized in the future. For example, researchers have identified that the genome contributes to your risk of developing a heart attack. This wasn't surprising. We knew that there was a genetic component to it. But what was surprising is that the risk was increased because an otherwise normal cholesterol suddenly became dangerous. So knowing the information for each individual helps us to reinterpret what normal or what healthy is for each individual, and therefore intervene early, where simple lifestyle changes can already reduce your risk. Now, we also applied it in the viral space, specifically COVID-19. And this is because we know that the virus is mutating. When it crossed from its original host to human and spread from human to human, it picks up mutations. Now, these mutations can lead to the virus being more infectious, meaning it spreads easier, or it can be more pathogenic, meaning the disease outcome is harsher. Either way, this information is encoded in the virus's genome, and therefore we can identify which one of those mutations might have causal effect with variant spike. With this capability, we can reanalyze the world's genomic data on a regular basis to look for viral mutations of concern that may arise and that we need to keep an eye on. For example, the Delta strain originated in India and is now the world's dominant strain of COVID-19. It has 15 mutations that make it more spreadable and potentially change the pathogenicity of the virus as well. Now, mutations like this, especially in unvaccinated countries, will arise on a regular basis going forward. Therefore, it is absolutely crucial that we have the capability of monitoring it and interpreting the mutations with their functional base going forward. And this leads me to another application areas where APIs are perfect, namely the capture of medical data. Now, with COVID, the biggest data bank in the world is called GIS8, and they started collecting data from the very beginning. Now, way back in May 2020, the patient information was very sparse, meaning most of the samples were annotated with patient status unknown. In October 2020, GIS8 made that field mandatory, meaning that everyone that submitted a sample to GIS8 had to put something into that field. But that something was free text. Therefore, the information of different kinds of annotations were overwhelming. And this is exactly where APIs can help, because it can translate the meaning between one data set and another. Specifically here, it could be a FHIR-based terminology, where everything is mapped onto a clinical terminology that is defined. And for that, the OntoServer can help. OntoServer is FHIR-based and it enables syndication for clinical terminology. It supports the advanced use of SNOMED CT and underpins UK's national health system. Therefore, it can broker the information exchange between silos of large medical data. And this is specifically in the genomic space necessary because the data is growing exponentially. Now, Frost and Sullivan estimated that by 2030, 50% of the world's population will have been sequenced, which means that the data that is produced in that space is larger than the traditional big data disciplines, YouTube, Twitter, and astronomy combined. And therefore, we need to think how we analyze the data, specifically embracing cloud-native technologies. So here I'm talking about serverless or function as a service. And this is how I think about it. Now, we're all familiar with desktop compute, where the focus is that you have full control over what you're going to install and how you're running it. But the downside is that you really have to look after your system. It's like owning your own car. You can go wherever you want, whenever you want, but you have to take that car to the service. Plus, you only have that one car. If you need something bigger, well, bad luck. So changing that, you could do by hiring a chauffeur. That person could come with a different car and can take the car to the service, which takes all of that off your hand. And the equivalent of that are auto-scaling groups in the cloud. But similar to a chauffeur, those things are really pricey because those auto-scaled groups, they don't simply go away if you don't need it in that instant anymore. And here is where serverless comes in. You do have the flexibility and no overhead, but at the same time, you don't have to pay the price for all of this. The systems scale up and down instantaneous, and therefore I think about this like a ride-sharing app, where you can demand the car right here and there, and if you don't need it anymore, then it goes away. We brought this flexibility to the genomic space, specifically for the exchange of genomic data. So in clinical genomics, it's quite common to look up a specific location in the genome in a large cohort. Say, for example, you have a patient that has a suspicious mutation in a particular gene, and you want to confirm that this mutation is truly deleterious. Therefore, you look it up in the large volumes of data around the world, and if a lot of people have that particular mutation, chances are it's not that bad. But looking up this kind of information in the world's data is quite complex. And here is where we brought in serverless technology. Rather than requiring that all of that data is consolidated into a single database or a set of single databases that are connected, we don't require that. For us, the data can stay in the individual files somewhere in the world, and all you have to do is register that particular file with the system. And we achieved that with serverless technology. Now, I'm not going to go through the architecture itself. Suffice to say that the data is located on an S3 bucket, the individual computes is handled with Lambda, and then all of that is brought together by an API, the API gateway namely, which brokers a standardized way of having the client interact with the data without having to give out the actual information, the private information of the patient sample. Doing it in a serverless way not only allows the flexibility, but it also makes it more cost-effective. Specifically, in the traditional way, it would cost an organization $5,000 a month in order to share genomic data. But we brought that down to $15 a month, which is less than a cup of coffee per day in order to contribute the knowledge of that particular organization to the world. We hope that this enables more organizations around the world to light beacons and therefore contribute their data to clinical research. Using the API gateway in such a way enables two analysis engines to talk to each other. And this, we think, will enable population scale systems, where you might have genomic information from everyone living in that country, say for example the US, which will be 350 million samples. Each one of those would contribute their whole genome, which is 3 billion. So in the end, you will have to handle 1 quintillion data points. And we think that serverless beacon and cloud-based technology can handle that. This brings me to the second story. Now that we understand the genome can be manipulated in order to develop advanced therapeutic applications. Jennifer Doudna, who received the Nobel Prize for her CRISPR work, has once said that the world around us is revolutionized by CRISPR, whether we're ready or not. And I think that is a very telling quote, because CRISPR already plays a role in a wide range of applications, from designing model organisms for clinical research to large-scale screens in order to understand how the genome works, biosecurity application to reduce the spread of invasive species, to gene therapy for handling genomic diseases or cancer. CRISPR can provide all this capability because it can be programmed to identify a specific location in the genome. Think of it like a mini postman, where you give it a specific address in the genome and it delivers the cargo directly to that address and no other address. Now, the critical thing here is to define this address uniquely. So it is truly only this particular location that you want to target rather than other genomic locations that you might not want to target. Defining this address is hence like finding a grain of sand on the beach. It has to be the right size, the right color, and the right texture for the machinery to actually bind with it and find. So you have to basically take each grain of the sand, which will be each locations in the genome that it could bind, and evaluate whether this one has the right properties, but also is unique enough. So therefore, not only do you have to touch all grains of the sands once, you have to touch them again in order to identify that they are truly unique. This is computational, very expensive, and we do need to come up with new analysis strategies in order to handle this. We specifically work with the Children's Medical Research Organization here in Australia in order to treat a liver disease. Here, the idea is that the CRISPR editing mechanism is packaged up into a envelope that goes to a specific tissue type, in our case, liver, and therefore within those liver cells go to a specific location in the genomic position and correct the misspelling that leads to a genetic disease. But in order to bring gene therapy to the clinical practice, there is a long road ahead of us. Not only do we need to know what kind of tissues and what kind of genetic misspelling is involved in the disease, we also need to develop something that is specifically targeting the tissue and not going to other tissues or organs. It needs to go to a specific location rather than hitting the genome broad spectrum. And it also needs to be safe for the individual, because every mutation that you have can change how the CRISPR interacts with your genome. From there, of course, it needs to go into clinical trials in order to prove that all that theory is actually working out in practice. We are focusing on a number of areas in this value chain in order to improve or make it possible that gene therapy can go to the clinical practice. Now, for all of those, APIs are again crucial, because as we established, it can broker the information exchange between data and the analytics, but also it makes it easy to exchange the analytics with a new version of the analytics that is based on the latest research or updates from the clinical practice. Built on this principle of modularity, we have created GTScan, which is a cloud-based platform like a search engine for the genome, where researchers and clinicians can type in the gene that they need to edit, and it comes up with the best strategy for it. In fact, GTScan was the first serverless technology to prove that this cloud-native approach can be used to cater for complex workflows like gene research. By exposing all of the analytics capability of GTScan in an API, we're able to do really complex but automated analytics running on top of it. So for example, we developed a notebook that hits the API with certain questions. For example, can you find target sites that are specific to heart compared to other organs in the body? With this, we can develop targeted treatment that are only going to be active in the heart where it is needed. Similarly, this capability allows other questions to be asked. For example, can the CRISPR differentiate between one virus and another? Say, for example, one COVID-19 strain that is highly pathogenic compared to a baseline, a normal version of the virus. Therefore, next time when you go through an airport security check, maybe there is a CRISPR diagnostics panel, that's what it's called, where there's a panel with lots of CRISPRs that are specifically programmed to identify individual viruses and differentiate between them. Now this brings me to the last story of how to modernizing collaborations. I would argue that cloud, and by extension APIs, really made it possible that solutions from one end of the world can be used on the other, and therefore access to talent is much easier, and information between academia and industry can flow easier. To demonstrate this, we have put VariantSpark, our machine learning method for genome analysis, onto the AWS marketplace. In fact, it was the first product, the first health product from a public sector organization to be on a digital marketplace. So if you're not familiar with marketplaces, you can think of them as sort of an app store. But rather than installing a specific app on your mobile phone or tablet, it does the same thing in your cloud account. And it does that by having the architecture, the workflow, the data handling implemented in one file. And this file then stands up automatically everything in your cloud account. Specifically for genomic research, this solves a lot of problems. It makes research more reproducible because the hardware, the workflow, and everything is exactly the same whether you do it in one lab or another lab. It also helps with the privacy. Data does not have to be transferred out of one account into another in order to be analyzed. The analysis is going to the data. I therefore would call these digital marketplaces some sort of API because it allows the information exchange between one computer and another. So with all this technical capability and the need of actually having good interfaces to work together in the digital health space, why is it that there is no good solution yet? Well, as API experts, you probably know that the solution is only as good as the people agreeing to work with them and working together. And with researchers and clinicians, not IT experts, it is very difficult to actually come up with a solution that is purpose-built, beautiful, and fit for purpose. So as the experts in that field, I would encourage you to reach out to a clinician or a researcher and see if there's anything that you can help in order to provide your expertise of bringing the aim of working together one step closer. I therefore think this will revolutionize how we do collaborations in the future. It would change how we think about individual systems and how they can be modularized, built upon, in order to create better solutions together. So the 3 things to remember from my talk is that bringing genomic information to the clinical practice is built on APIs. We do need to bring the analysis to the data, and we've done that with VariantSpark on the digital marketplace. Similarly, data needs to be exchanged with the world, and we've done that through the serverless beacon and digital serverless technologies. We also contribute towards exploring new and advanced treatment avenues going forward, specifically gene therapy-based ones. For this, we developed GT-Scan on this concept of modularity, where new information can come in. The whole system does not have to change, only individual elements can change, and this again is handled through APIs. But most importantly, the way we do collaborations in the future, I think, needs to change. We need to work seamlessly together with academia, clinic, and the industry. And I think cloud technologies, and by extension APIs, will really help bridge that gap. If you want to find out more about the research that we do or the architectures that we've built, please visit our webpage. On there, we have examples of how we're using machine learning in the clinical practice, as well as other application areas. And if you have ideas on how to improve the APIs and solutions that I presented in this talk, please get in touch. I would love to hear your feedback and suggestions on how to use cutting-edge technologies to bring the domain knowledge into the clinical practice and improve healthcare as we know it. With that, thank you very much for listening and talk to you soon. Bye.